The overall goal of this co-culture protocol is to measure the effects of ionizing radiation on Caco-2 epithelial cell monolayer permeability and tight junction function in the presence or absence of peripheral blood mononuclear cells.
This method can help answer key questions in the radiation biology and radioimmunotherapy fields about possible synergistic effects between ionizing radiation exposure and immune cell functions.
The main advantages of this technique are that different stimuli and cell population can be tested and observed phenomena uncoupled through ad hoc complementary measurements.
The radiation procedures aims at considering the Caco-2 cell monolayer as tumor volume irradiated with the appropriate beam and accurate dosimetry, as is case of radiotherapy on patient.
One week before their irradiation, seed 5 × 10⁵ Caco-2 cells in 2 milliliters of complete medium into 1 sterile 1 micrometer pore diameter cell culture insert per well in a 6-well cell culture plate. Add 3 milliliters of fresh complete medium into the bottom compartment of each well and place the cells in a humidified cell culture incubator at 37 degrees Celsius and 5% CO2 for 7 days. On the last day of the incubation, add 25 milliliters of Ficol to a 50 milliliter conical tube and carefully layer 25 milliliters of freshly collected whole blood over the Ficol. Separate the cells.